Well, I'll be doing a presentation on genetics and inheritance, and it's going to be a three-part series. Well, my focus areas are the following. I'll be looking at part one, which will delve deep into monohybrid crosses with complete dominance, as well as monohybrid crosses with incomplete dominance, and then also with codominance, and I'll end with sex determination. This will continue with part two, where I'll be looking at inheritance of sex linked diseases. And then we will be looking at multiple alleles, example, blood grouping. And I'm going to end it with part three with a focus on pedigree. So let's start by looking at the site of meiosis because genetics involves all the topics. We have to understand that meiosis is a cell division that usually results in the formation of sex cells. Sex cells are also called gametes or reproductive cells. In plants, meiosis occur in the male part called the anther to produce pollen grain, and then in the ovary to produce ovule in the female part. In humans, meiosis occurs in the testes of males to produce sperm cells and in the ovary of the females to produce an ovum. Pollination is a transfer of pollen from the anther to the stigma. And then after pollination, the sperm in the pollen fuses with the ovule to form a zygote in a process called fertilization. Well, we have to know the man on the screen. He's regarded as the father of genetics, and he's called Gregor Mendel, an Austrian monk. Uh, he he was a monk, a type of priest, and he is regarded as the father of genetics because of his work on garden pea plants that helped significantly to explain how genes are passed from parents to offspring. So we look at Mendel's experiment with his garden pea plants. He noticed at least seven traits or phenotypes that appeared to be inherited. So these were the traits he observed. One, the seed shape, the seed color, the pod shape, that is the container holding a plant's seed, the pod color, flower position, whether it is axial on the side or terminal on the top, and lastly, the stem length. So here are the traits observed by Mendel on your screen. Now, we are now going to test Mendel's experiment. So, from the experiment he did, he observed that garden peas appeared in at least two types of heights. We have tall and short. And since peas are self-pollinating, that is self-fertilizing, tall peas appear to produce tall plants, and short peas produce short peas. Mendel's first genetic cross involved tall peas crossed tall peas crossed with short peas, and he decided to take a short pea plant, and then he crossed them with a tall pea plant. And then these are the things he observed. These are the results. In the first group of offspring, that is the first generation, the F1 generation, the cross yielded or produced only tall peas. That is, all the peas were tall. And then he later on took the F1 peas, that is the new tall plants, and crossed them with themselves. That is interbreeding. And then doing so, he produced the second group of offspring. That is the F2, the second generation. And these were the results. Some were tall, others were short peas, and they were in a ratio of 3 is to 1. That is, uh, about 70% of them, 75% of them were tall, corresponding to 3 tall, and 25% or 1 dwarf. Now, this will take us to the types of genetic crosses. As we know in the curriculum, there are two types of genetic crosses. We have monohybrid crosses, which is a type of genetic cross that involve only a single or one characteristic or trait. Remember, the word mono means one. So that should be an easier way for you to remember. Dihybrid crosses. The word di means two. It is a type of genetic cross that involve two characteristics or traits. Now let's explore Mendel's uh, laws and its application in these two types of crosses. So we're going to apply Mendel's law there, and now we're going to start here. So we all know monohybrid cross refers to a genetic cross involving only one characteristic, sorry. And so we have on your screen, we have two different types of the letter S. One is uppercase, one is a lowercase. The uppercase is called a dominant S, and then the lowercase is a recessive S. So I'll not be using the terms recessive and dominance. So the dominant produces the smooth color or makes the seed or the organism smooth, and then recessive is for wrinkled. So if we have such a combination where we have two letters there, meaning after fertilization we have um, a diploid condition where we have two letter S. One came from the father, one came from the mother. If they are in this combination, then that becomes the genotype of the organism. So the genotype of this organism is homozygous dominant. Homo means the same because we have two same letter S standing side by side. And according to the key, the dominant is for smooth, and then the recessive is for wrinkled. So if you have a homozygous condition on your screen like this, that will produce a phenotype that is smooth. We have to know it is a genotype that determines the phenotype. The phenotype is what we see, is the physical appearance. So the two letter S represent the genotype, and then the smooth uh, appearance represent the phenotype. The smooth is determined by the genotype, which are the two dominant S standing together. Now, over here we have two combinations of S. One came from the father, one came from the mother during um, fertilization. That is, this zygote here is diploid. Di means two because it has two sets of alleles. One is dominant, one is recessive. And this zygote inherited one allele from the father and the other allele from the mother. These two alleles forms the genotype, and the genotype determines the phenotype, what we see and feel and touch. That's the physical appearance. According to the key, dominant uh, is for smooth, and a dominant allele, which is a capital letter, it blocks the recessive allele from appearing, from showing. Hence, the phenotype is going to be smooth because the small letter s, which is recessive, was blocked from expressing itself. Hence, the overall or the resulting phenotype will be smooth. Now, we have two letter s here again. Both of them are small letters. And again, one was inherited from the father and another one was inherited from the mother during fertilization. And it forms the genotype of that organism. Because they are two and they are the same, we use the word homozygous. But then you can see both of them are small letters. Hence, if they are small letters, we use the word recessive. Therefore, we call this combination homozygous recessive. Now, because this forms the genotype, and also because the genotype determines the phenotype, the resulting appearance or the resulting phenotype of this organism will be wrinkled. Why is organism wrinkled? Organism is wrinkled because there is no dominant allele there. And organism is homozygous recessive, having two recessive alleles for wrinkle. So that is why the organism will be wrinkled. Then we go on to a situation like this. So let's look at this situation and let's use simple explanation for it. A man with genotype, that is two dominant letter C, that is for curly hair, and a woman with genotype, that's one capital C dominant and one recessive, also for curly hair, have a child. Explain why their child cannot have straight hair. So on your screen, we know the dominant letter C is for curly hair and the recessive C is for straight hair. So let's look at the explanation. Why would their child not have uh, straight hair? It is impossible. The child cannot have it. And then the answer is this. The C allele for curly hair is dominant as it is given to you in the question. And the small letter C for straight hair is recessive. So you have to tell us which allele is recessive, which allele is dominant. This is to enable us to know that you understand the question. Then the father is homozygous dominant with two letter C. So he will always pass on a dominant allele letter capital C during fertilization and or inheritance. The mother is heterozygous, and that is she has one capital C and one small letter c, that is one dominant allele, one recessive allele. So she can pass on either of them. She can pass on a dominant allele for curly hair or a small letter c for straight hair. The possible combination for the child are here. The child will either become or carry two dominant allele, making making it homozygous dominant, or the child can either be heterozygous. In both cases, the hair will be curly. That is, if they have to fertilize uh, the gametes. Therefore, the child cannot have straight hair since both possible genotypes result in curly hair. And that's how simple uh, it is in the explanation. The explanation is based on fertilization. You can only contribute what you have. You are forming a child, so you have to contribute your gametes, and your gamete carries your genes. You can only pass on your genes to your offspring, and what you have, that's what your child might carry in half. So when you're explaining, you have to think in that direction that you are passing on an allele that you have into your child. Half of it is going to your child, and half of your partner is also going to your child. So it is the fertilization of both gametes that will result in the child's phenotype. This usually carries 5 marks, so you should practice how to go about it. All right, now let's go on to another situation here. State the phenotype for this genotype. So we have this genotype that is heterozygous. We have capital dominant A and recessive A, and explain why. So here, capital A is for brown skin. That's dominant A is brown skin. Recessive A is white skin. So the phenotype for the genotype, that is capital A, small letter A, is brown skin. We have decoded it. What are our reasons? This is because the capital A for brown skin is dominant over the small letter A allele for white skin. Now you don't have to write small or capital in exam. Just simply put capital A or small letter A, and we know what we are trying to say. Since dominant traits are always expressed when at least one dominant allele is present in the genotype, an individual with capital A and small letter A will have a brown skin. Simple explanation. So you have to use the definition of either recessive allele or dominant allele to explain. So remember the person, it has been given to you the capital A is for brown skin. Now the next point, which was this one, okay, which is this one, was a definition of the word dominant allele. It was the terminology. It was actually to define it, and then you draw your conclusion. Therefore, an individual with capital A small letter A will have a brown skin. This will carry about 4 marks because you have to state the phenotype. It can even be 5 marks. Then you can go on to say, even though the person carry a recessive allele, you can also explain why the recessive won't appear. Now, the reason why you're going to explain why the recessive won't appear is in the definition of the term recessive allele. So the recessive allele is only expressed when in the homozygous state. So that is why it will not appear. A woman with genotype uh, capital B small letter B for black hair and a man with genotype homozygous B, black hair, have a child. Explain why their child cannot have blonde hair. It has been given to you. Capital is for black hair and small letter is for blonde hair. So the answer is still the same like the previous one. Remember, you have to tell us which one is predominant and which one is recessive. So the capital B allele for black hair is dominant and the B for blonde hair is recessive. Just by telling us which letter is dominant and which one is recessive gives you a mark or two. You can get 2 marks here. And then you go on to explain what the parents carry or the genotype of the parent. So the father is capital B, has two capital B, is homozygous dominant, and will always pass on a B allele. The mother is heterozygous, so she can pass on either B, black hair, or B, small letter B for blonde hair. So the possible combinations for their child are that's homozygous B for black hair or heterozygous B for black hair again. Therefore, the child cannot have blonde hair, as both possible genotypes result in black hair. And that is how you explain in exams. So let's look at this one also finally. If capital R is for red and small letter r is for white, predict the phenotype for RR, two small letter r, and explain why. So the answer is white flowers. Reason R, that is capital R is dominant over small letter r, which is white. That gives you a mark. That is 2 marks there. This means that for the flower to be red, it only needs one capital R, which is dominant. The genotype RR because two small letter r means the flower has two recessive alleles for white. No dominant R allele is present to mask the effect. Okay, since both alleles are recessive, the phenotype will be white flowers, as there is no dominant R allele present to produce red color. And that is how you answer it in short. Now let's explore Mendel's law again. Remember we are looking at dihybrid, which involves only two characteristics. And so we have, remember we have done previously the monohybrid. Now let's look at dihybrid cross here and let's look at the situation. So over here, we are given two sets of letters, the G's and the S's. So we have two types of G, we have two types of S. We have the capital G, which is dominant for green, and we have small letter g, which is recessive for yellow. And then we have another type, two types of S, dominant S for for smooth, and recessive S for wrinkled. So if you have a combination like this, that is under the G, we see we have two genes here. It means one came from the father, one gene came from the father, one gene came from the mother. And then this one deals with color. This allele, the genes, they deal with color. And there are different kinds of color. Here we are giving green or yellow. Then the S deals with texture. that is smooth or wrinkled, and that is a different phenotype. So again, the two S, one came from the father, one came from the mother. And as you can see, we are looking at dihybrid cross, which involves two characteristics or two phenotype or traits. And the phenotypes here are color and texture. Color is controlled by gene allele, and texture is controlled by S allele. Under each allele, some are dominant, some are recessive. So if you have this combination here, it means one gene came from the father, one gene came from the mother, and then again one S came from the father, one S came from the mother. There are thousands of characteristics that we can put together, but we have only four of them on your screen. Half of them coming from the mother, two coming from the mother, two coming from the father, because we are not able to put write all the letters on one screen. That is why we chose only two maximum, what we call dihybrid. You can even have trihybrid or tetrahybrid. You can continue as many as you wish or as you like, but then for curriculum's sake, we are sticking to only two characteristics where we're looking at two phenotypes, color and texture. So based on the key, since the capital G is for green and capital S for smooth, the phenotype will be green and smooth. Then we have another combination here. We have two types of gene, two types of S. The G, one is dominant, one is recessive. The S, one is dominant, one is recessive. So also under the G, one came from the father and the other gene came from the mother. The S2 is the same. One came from the father, one came from the mother. So what will be the phenotype? Here, if you look at the gene, you can see it's heterozygous. One is dominant, one is recessive. And remember, the dominant is always expressed in the phenotype, whether it is homozygous or hetero. Hence, the phenotype for this genotype will be green. The S from the chart, from the key, the dominant is for smooth. Hence, the phenotype will be green and smooth. And if you do the same, guess what the phenotype will be? You're going to have green. And you can see we have two small letter S, meaning it's going to give us wrinkled. And then the next one, we have two small letter G, and you know small letter represent recessive, which is for yellow, and two small letter S, which is for wrinkled. So the color will be yellow and wrinkled. And then we have here two small letter G's, which is homozygous recessive for gene. That will make the color yellow. And then for the S, it is heterozygous. One is capital, one is dominant, one is small letter recessive. So the color is going to be yellow, but then the texture is going to be smooth. And then that is how you work it out. All right, so this is how you interpret it for 2 marks. Yellow is 1 mark, smooth is 1 mark, green is 1 mark, wrinkled is also another mark. So make sure you know these things. You treat the letters separately, the G on one side and the S on one side, and then you deal with them. You interpret them using the normal genotypic interpretation approach. Now, let's look at how to generate the gametes in dihybrid crossing. So let's start here. We're going to generate gametes in dihybrid, something which is often asked in exams. So let's say you are given this genotype of one parent, and then you have to generate the gametes. Okay. Remember, gametes are haploid cells carrying half the number of chromosomes in the somatic cells. So what we have on your screen is a somatic cell, is dihybrid, is diploid, sorry, and now we need to produce the haploid condition of it. So this is how you generate the gamete. So you start this way, you start with the first letter H. I'll be dealing with the H because the H is on my left. We read letters from left to right, so I'll be dealing with the H. I'm going to use the H to help me generate the gametes. Using the D is possible, but it might be confusing. So I will advise you to start with the H. So whilst I stand with H, I draw my arrow. The arrow touches D. Let the arrow not touch H. H is not crossing itself. All the H will have to cross all the D's. So the H will cross this D. That will give me the first gamete. That is haploid. We have 2H, 2D, but this gamete consists of 1H, 1D. That is half of what we have on your screen. So 1H is half of the 2H, and the 1D represent half of the 2Ds. And then we continue again, still with the capital letter H, we produce the next one, and there we have it. So you just draw your arrow on the question paper, and then you can get your first two gametes. Now I'm done with the H, capital H, there's nothing to cross again. So I go to the small letter H and I do the same. Yes. And then good. It's better to start with the H on one side and the D's on one side. It doesn't matter which one comes first, but it's advisable to start with the H, whether it's a small letter H or not, start with it. And then we do it this way. And then what do we have? Yeah, that's what we have. So this is how we produce the gametes. Now I have the gametes, and I'm gonna get my 2 marks just for doing that. Okay. All right. So then we go here. Yeah. Again, now we're going to do it in reverse. Now we'll be given the gametes. Now you have to produce the genotype. So we are reversing the process now. So let's see how you do it. So you put all the capital letters in one box. So all the H in one box, two becomes one. And then all the D. Oh shit. Okay. All right. So that is it here. Sorry for the error. So all the H on one side, that is it there. Now the next one will be the D. Very good. So the 2D became one. The two small letter H also became one. And the two small letter D became one. Sorry for the glitch. So that is it. Two becomes one. And then what we have here is what is called the genotype. So in exam, sometimes they will say use the gamete to produce the genotype, or use the genotype to produce the gamete. And that is how you do it. Okay, we have another situation here. You have the gametes here. Now you're supposed to produce the genotype. Let's see, two becomes one, and then this two becomes one, and then that two becomes one, and then you have this way, two becomes one, and you have it that way. And yes, and that is how you produce the genotype. All right, let's continue in our next slide. All right, so let's look at a situation here. We all know that hybrid cross is a genetic cross involving only two characteristics. As the name suggests, di means two. So we have the key here. Capital G is for green, which is dominant, and small letter G, which is recessive, is for yellow. That means the yellow phenotype is recessive. The green phenotype uh, is dominant. Same applies to the S. Capital S is for smooth, making smooth dominant phenotype, and small letter S is recessive, producing a phenotype that is wrinkled. So here we are being told heterozygous green and wrinkled, and heterozygous green and heterozygous smooth. All right, so we have two parents. One is heterozygous green and wrinkled. The other one is heterozygous green and heterozygous smooth. So let's produce the genotype for these two parents. Remember, the first parent has a heterozygous green genotype, a phenotype, right? That means green is heterozygous. There are two ways of being green. Either you are controlled by homozygous allele or homozygous genotype or heterozygous. So over here, we're going to have two genes. One is capital, one is small letter. There's only one way you can become wrinkled. That is when you have two recessive alleles. So guess what? The other parent is also going to be uh, having similar one for the gene, same for gene, but then for the smoothness, remember there are two ways of being smooth. Either you are homozygous or heterozygous. But here the statement is heterozygous. Hetero means different. So that is why we have the genotype here. If you get the genotypes wrong for the parent, everything will be wrong. The entire concept of genetics is based on your interpretation of those 10 terminologies: dominant, recessive, heterozygous, homozygous, alleles, and genes chromatin and chromosome. Those are the words you are going to be playing with in exams. So this is our table. Now we have to generate our gametes. So we have to use the same method that I showed you previously. Use the arrows to produce the gamete. If you start with the gene, the capital G, you draw an arrow to S, you're gonna get that. You're gonna draw another arrow to S. And I'm done with the capital G. You stand with small letter G at the bottom there, and you draw an arrow, and that will give you this. You draw another arrow to the second one, and you got it. Now let's do for the same parent. You can give it a try. Try and produce the gamete for this parent genotype. You stand at G, you draw your arrow, and that's what you get. Then you are left with one S to go. You draw another arrow, and then you get that. Now you are done. There's nothing to cross. Gene cannot cross gene. The gene crosses with S. So I'm done with the capital G. I go to small letter G. I draw the arrow from the bottom. I do it this way, and I do it that way. Now that I have the gamete, what do you do with gametes? Gametes refers to sperm cells and ovum. What do you do with them? You fertilize them. So I'm going to show you the first line only, and then you can try the remaining ones on your own. So this is it. Remember, it was the GS, the capital G and the small letter G, the capital G and the capital S that join together. So here you put the G's on one side and the S's on one side. And you do the same thing here. You cross them, G's on one side, S's on one side. Now you must be able to interpret the phenotype of these zygotes or the offspring. So the first one is going to be green and smooth. The second one is going to be green and wrinkled. And then it continues this way and continues that way. So this is how you do it. You can finish up with the rest, which is key and very important for mastering the concept. Good. So now let's go to the three laws of Mendel. So according to Mendel's laws, we have the following here, where we are looking at the law of segregation, the law of dominance, the law of independent assortment. So these laws, mostly in exams, you have to quote them. So what does the first law say? The law of segregation. It states that during gamete formation, the alleles segregate or separate from each other so that each gamete carries only one allele for each gene. So that is the law or the principle of segregation. Segregation means to separate. And this thing occurs during gamete formation. What is one single word for gamete formation? The word for gamete formation is gametogenesis. And you know, gametes formed by meiosis. So another word for gametogenesis is the word gamete formation. What about the law of dominance? It states that in a heterozygous genotype, one possessing two different alleles for a particular trait, one allele will be expressed over the other, masking or blocking its effect, meaning the dominant allele will be expressed over the recessive allele, and it will do so, that is, the dominant allele will do so by masking or blocking the effect or the expression of the recessive. Now, the last law is called the law of independent assortment. The word assortment means to distribute. This law states that during gamete formation, alleles separate and therefore assort independently of each other during meiosis. It is highly possible you can be made to quote for 3 marks, or you can be made to explain how it operates. Now you have to use those words: independent, assortment, dominance, segregation. You have to use the meaning of those words when you're explaining. And all these things are happening during gamete formation, that is gametogenesis, where the alleles separate and they assort. Assort means they distribute themselves of each other during meiosis. And you know, meiosis is what makes gametogenesis possible, or gametogenesis occurs by meiosis. Now let's look at it in real life. So in real life, when applying Mendel's laws, this is what we have here. we have two parents. One is tall and one is dwarf. So we have parents. We have the following parents here. One parent's genotype is heterozygous. The other parent is um, homozygous recessive. So let's see how Mendel's laws apply here. You can see over here during gamete formation, the the two letters, capital T, small letter t, they were separated. Same thing applies to the dwarf with the genotype, two small recessive T or two small letter T's. You see that they have been separated by the arrow, and it was because of the law of segregation. Segregation means to separate. So now you have separated the alleles. Then we look at it here inside the circle. When the letters separated, they entered it. In other words, the letters separated so that they can enter those circles. Those circles represent the gametes. The gametes can either be a sperm cell or an ovum. So that is during gametogenesis when these gametes are forming, the alleles, okay, the different types of genes in the individual will separate themselves or segregate, and then they will individually or independently enter into a sperm cell or an ovum. That is, the ovum will carry the gene from the individual, or the sperm cell will carry another gene, another allele from the individual. Now, the sperm does not choose which gene it should actually receive. Whatever will enter it does so on its own accord. It is independently distributed. It is done not by the will of the individual. Now, sometimes you have a situation where we have two combinations, such as we have on your screen under the F1 generation, where we have a capital T and a small letter t, and the other side we have two small letter t. That one won't be a problem when you have two small letter t, because you know what the outcome will be. It's going to be the same as the information in the letter. Here it's going to make the organism appear short or dwarf. But then when you have two different alleles, one is dominant and one is recessive standing side by side, who will determine the phenotype? Will it be the dominant or will it be the recessive? To solve that problem, like this one, we apply the law of dominance. Only the dominant allele will be expressed in the phenotype. So, according to the second law of dominance, it states that in a heterozygous genotype, that is one possessing two different alleles for a particular trait or phenotype, one allele will be expressed over the other. And the allele that will be expressed over the other is the dominant allele. And it will express itself over the recessive allele. And it will do so by masking. Another word for masking is the word blocking. So the dominant will block the recessive from appearing or from expressing itself. So you have a situation like this: capital T for tall, small letter t is for dwarf. What will be the end result if you have a zygote with two alleles? One is dominant and one is recessive. And where did this zygote get the two alleles from? One came from the mother, one came from the father. Maybe the capital came from the father and a small letter came from the mother. Hence, the organism will be tall. Why would the plant be tall? The plant is tall according to the second law of Mendel, which is the law of dominance. So the letter T, that's capital and small letter T, okay, and the tall plant, what do they represent? The tall plant represents the physical appearance, which is the phenotype, and then the two letters standing together, they must always be two when is because okay, there must always be two, and I don't want to say that thing now. There must always be two because it is diploid, and one T came from the mother, one T came from the father, and that is what is called the genotype. That's the genetic makeup. All right, now let's look at the types of dominance. So we have three types of dominance. Check very well. We have complete dominance, incomplete dominance, codominance. Very important. So let's start with complete dominance. So with complete dominance, the dominant allele completely masks the recessive one. That is, we have a monohybrid cross with complete dominance. We have capital R for red, small letter r for white. Here, in this situation, only the dominant allele is expressed in the phenotype. And the dominant allele will do so by masking or blocking the effect of the recessive one. So we have homozygous red flower and a white flower. What is the F1 generation? You can predict. So with complete dominance in a homohybrid cross, capital P is for purple, small letter p is for white. So we have two parents. One is homozygous purple, the other one is just white. Why is the white not homozygous or heterozygous? Because there's only one way of producing white. For it to be white, you just have to be recessive because white is a recessive phenotype controlled by a recessive allele. Hence, homozygous purple will appear this way, white will look like this. Now, when they cross, okay, uh, the one parent will present a sperm, another parent will present an ovum. A sperm and an ovum are haploid. And on your screen, we have two letter P for purple, and the other one, two small letter P for white. This, since they are two, we call them diploid condition. So for you to have a child, one must donate half of whatever he has. The offspring is gonna look like this in terms of genotype. We have two letters here. One is capital, one is with small letter. The capital came from one parent who is homozygous. The small letter came from the other parent, which was homozygous recessive. Both of them are homozygous. One is dominant, homozygosally dominant. The other one is homozygous recessive. So for this generation, for this offspring with two different alleles, one dominant, one capital, one recessive, small letter, where did it get it from? It got one allele from the purple flower, it got the dominant from the purple flower, and got the small letter from the white flower. And that is it. Both flowers presented half, not the two. You just send half of whatever you have. So sending half of whatever you have is what is actually done during gametogenesis. That is when you contribute half of your life, half of yourself into your child. So half of what you have is what will join together in the zygote to turn the zygote diploid. So the color will be purple. For incomplete dominance, we have a situation like this. We have the presence of intermediate or range of traits or phenotype. So we have a situation like this, and both alleles are dominant, and none of them is being expressed. Both alleles blends together, or they mix perfectly in the phenotype. So instead of the child resemble any of the parent, the child won't. Like we have on your screen, on the right side of your screen, two parents, one is red, one is white. They produce an offspring. The offspring doesn't resemble any of them. One parent might deny that the child belongs to them, or maybe one will accuse the other one of cheating. But it cannot always be the case because the offspring is pink in color. And how do you produce pink? It is when you mix red and white perfectly. So what we have here is a blend of two phenotypes, red and white, producing that color. Same thing that can be applied to height. So we have dominant T for tall, dominant for dwarf. So both alleles are dominant. What would be the phenotype of their offspring? Will the offspring be tall or dwarf? According to incomplete dominance, the offspring won't resemble any of them. The offspring will not be tall, it won't be dwarf. So it's going to be a mixture of the two. When you mix tall and dwarf perfectly together, what do you get? You're going to get medium. So that is what incomplete dominance is. This is actually doesn't follow the natural rules, hence it doesn't fall under the natural Mendelian genetics. It's actually a unique case. Another example is here. Neither allele is dominant. One parent is red, one parent is yellow. They produce an offspring that doesn't resemble any of them, though they share similar phenotype. Both of them, all the three are actually appear as a flower. And if you look at it carefully, it's a family of three with three different colors. So here, when an organism is heterozygous for a trait, it will show a third phenotype, and that is it. It's because neither allele is dominant And there's a third phenotype in the family. We have red, we have yellow. Then they produce a third child, and the third color is orange. How do you produce orange? You can't deny that it's your child, or you can't accuse the other one of cheating. So you ask yourself, how do you produce orange? When you mix red and orange perfectly, you're going to produce a third color or phenotype, which is a perfect blend or a mixture of red and yellow, or as the other two. And this will result in the orange color. Now the third one, which is the last one, is codominance. Codominance here, um, like in the word co means like they're cooperating, like nobody is on top of the other. They equally understand each other. So over here, again, both alleles are dominant and unique. You can see we are not representing them the way we usually do. Like you start with red, with you start at R, the second one should have been R. Instead, it became W. And the reason is because both phenotypes are dominant and it's a different type of genetics. So here, both alleles are expressed equally in the phenotype. And we have a situation like it is on your screen. Two parent, one is all white, the other one is all red. They produce an offspring. Now, because both alleles are dominant, the offspring start showing the phenotypes of both parents, and it shows the child belongs to them, and you can deny. So if you have a black fair cat and a gray fair cat, and they cross and they will produce an offspring. What will be the color of the offspring? According to codominance, the offspring, the cat or the kittens will not be black, neither will they be all gray. It's gonna be a perfect combination of the two colors appearing at the same time. So the phenotype is gonna be black and gray. Okay, so let's compare the two types of dominance. We have incomplete dominance, which is a type of dominance where neither alleles are dominant over the other, and there's a perfect blend in the phenotype. So you mix red and white perfectly, you get pink. On the other hand, with codominance, uh, you have two alleles and both are equally expressed. So the offspring displays the phenotype of both parents equally. So you can see in the phenotype we have of the offspring, we have three red, three white. Neither is dominant over the other. They are equally dominant. So that is how both of them are rated. So in monohybrid crossing, we try to use the keys in doing genetic cross. So we have this. Okay, let's say that is your genotype. If you don't want to have an offspring, that's the offspring. And here we decide to cross heterozygous genotype together. What do we get? That means you see you're gonna produce white. So over here we cross purple and then white, and all the offspring became purple. Then we decided to cross purple and purple. Now we got three purple, one white. How did it occur? Remember the F1 is heterozygous. If you mix heterozygous and heterozygous, you're going to get three purple and one white. And this is how we got a white. The white came from here, you see. And then the first purple would have come this way, and then the second one would have come that way, heterozygous, and this one too is heterozygous. So if you mix two heterozygous phenotype together, you are going to get the ratio 3:1. Three of them will resemble the parent's color, and a missing color or a recessive trait will appear. From your screen, you can tell that purple is dominant because you mix purple and white disappear, or white is blocked, or white is masked. That means white is recessive. So why do we have a lot of purple? It's because purple is dominant. Why do we have few whites, or why did white disappear and reappear again in the F2? The answer is because white is recessive. Now, when you're explaining, you have to use the definition of recessive allele. So you have to say the white is only expressed when in the when it's in the homozygous condition, and when there's no dominant allele for people in the zygote, in the zygote exactly. So let's go to the next one, which is the last part of part one, sex determination by males. So here we need to know, in genetics there are two types of sex or gender: XX for female, XY is for male. That is if the letter X and Y is given to you. So when you are asked to explain how sex is determined, you can do a genetic cross to do it, or you can put it into writing. That is, if you can express yourself. So you have to know the father is a male, will carry, hence the genotype will be XY. The mother is a female, hence the genotype will be XX. Then you use the normal genetic cross to produce the gamete for both. Then you cross them. Then you find your F1. Then you check for your the genotype for the F1. Then you display the phenotype for the F1, and then you do your ratios, and you can be able to tell the probability of having a male child or a female child. In both cases and in all cases, it's going to be 50% probability of having a male child and 50% probability of having a female child. This is a karyotype, what we call a human karyotype. It is a display of the number and shape and arrangement of the chromosomes in the nucleus of a somatic cell. So in humans, we have 46 chromosomes. So that's our karyotype there. They are given positions and numbers from 1 to 23. Each number carries two chromosomes, and each chromosome exists in pair. There are two chromosomes at each position. And the reason why there are two at each position is because the two chromosomes are homologous, meaning they are similar in length, similar in structure, similar in size. They have the same gene locus. They have the same position of centromere. Their centromere positions are the same. And from position 1 to position 22, all the chromosomes are homologous. They are two each, and they are very similar in length, in size, in structure, and have the same gene locus, except position 23. Sometimes the chromosomes appear identical or homologous. Sometimes they do not appear to be identical. In males, their chromosomes at position 23 appear to be unidentical. Often, and at all times in males, there are two chromosomes at position 23. One is large, one is small. And when it's like that, the person automatically becomes a male. Or males ends up having such chromosomes. If a person carries two identical chromosomes at position 23, that person automatically becomes a female. So that is how you get to know your gender. So the chromosomes from 1 to 22, which are identical and hence homologous, are called autosomes. The chromosomes at position 23, which are sometimes identical in female and sometimes unidentical, like the case of male, are called sex chromosomes. And sex chromosomes are also called gonosomes. So you must watch out in exams. If they ask you what is the gender of the individual, don't be in a hurry to say the person is XX or the person is a female because of she has XX chromosome. You only say XX if it is given to you in the diagram. If there is no letter X and Y in the diagram, you have to observe the appearance of the chromosomes at position 23. If the chromosomes look similar in length and size, that is a female. So you normally say a female will have two large identical gonosomes. You only say that if the letter X and Y are not X and Y are not given, or if the X's are there, you can simply say a female will have two X gonosomes at position 23. So you don't use the letter, you don't say the female or a female will have two X chromosomes if it's not given to you in the exams. If you go and write that, it's going to be wrong because the letter X wasn't given. What about male? A male will have two unidentical gonosomes, one large and one small at position 23, full stop. If the letter XY is not given in the question paper, this is how you answer. But if the letter X and Y are both given, you can also you can continue to say, you can still say a male will have two unidentical chromosomes, one large and one small at position 23, which is correct. Or because the X and Y are given in the exams, you can also say uh, a male will have two unidentical chromosomes, X chromosomes and the Y chromosomes are pair 23, and that one too is acceptable. So you need to watch whether the XX or the XY is given or whether they are not given. If they're given to you, you can go ahead and say a female will have XS and a male will have XY at position 23. If the letter X, X, and XY, if those letters are not given, don't try and write it as an answer for male or female because it will be marked incorrect. You only mark what you see. The question will tell you from the diagram what is the gender of this person or this individual, or is this individual a male or female. From the diagram you are seeing, not what you think you know. So sometimes what you think you know can help. You have to follow instructions. Now, these chromosomes, position 23, are what is called gonosomes. So explain why there's a 50% chance of a male zygote being a male or female. And the answer is because male have only two sperms. Half will have 22 plus X chromosome. The other half will have 22 plus Y chromosomes. Depending on which sperm reaches the egg, there's a 50% chance of the individual being male and a 50% chance of the individual being a female. All right, so like I was saying, you need to watch and make sure you have um, you have information on whether those letter X's and letter XY are given in exams before you give an answer or you take a decision. So let's look at genetic or disorders in the gonosomes. All right, so here we go. Like I said earlier on, you don't see letter X and Y naturally in the cell of an organism. You're gonna see two chromosomes. One is tall, one is short, or one is large, one is small. The large one is the X, the smaller one is the Y chromosome. It is advisable to refrain yourself from saying males carry X and Y. You must stop using those because you don't see X and Y under the microscope. Rather, you have to describe what you see. This chromosome on your screen do not look like X, neither do they look like Y. One is taller than the other, or one is larger than the other. So that is how you describe it. It is important to take notes. Now let's look at the following terms. That's autosomal characteristics and gonosomal, also called sex-linked characteristics. You know, the sex chromosomes that determines our gender is called sex chromosome or gonosome. So what are autosomal characteristics? They are traits, phenotypes, or characteristics transmitted through the autosomes. Example, your height, color, these traits are transmitted through your autosomes. What about gonosomal traits? Gonosomal characteristics, they are traits, characteristics or phenotype transmitted through the sex chromosomes or gonosomes. Examples of such phenotypes are haemophilia, color blindness, and your gender also. All right, so this is how we represent the three distinct characteristics in exams. The first one we have autosomal. Uh, let's say we have phenotype. That's for brown color. And then we have to know the genotype behind the brown color. And that is what we have there. Just use the letters as it is, like we normally do in genetics. But when you are dealing with gonosomal or sex-linked characteristics, you have to be careful because, yeah, the phenotype appears the same, but then the representation of the genes uh, becomes something unique. And that is where mostly um, learners get it wrong. Instead of using the letter X, some don't. Some just put the letter X there and they forget to put the alleles on top. The X represent the X chromosome and the letters on top represent the alleles because the alleles are on the X chromosome. These are the genes on X chromosome. You don't find them anywhere. Mind you, you are dealing with gonosoma. So that's how you represent it. Sometimes learners will just put the X there, they forget the alleles. Some of them just use the alleles. They put BB there or whatever the letter is, E or G, ignoring that we are dealing with sex link. When it comes to blood group 2, it has its own unique representation. Remember the phenotype, in this case, we don't put words there. The phenotype we use for the phenotype we use letters like B, A, O, K, or AB. But the representation of the genotype is what is so unique. It's different from autosomal genes and gonosomal genes. So over here, use the letter I. Isoagglutinin. You're not supposed to worry yourself about the letter I and its meaning. So for blood group, it's so unique. You put the letter I, capital letter, if it is a dominant allele. Dominant means B, okay? And in blood group, B or A. But in blood group, we don't have a recessive allele. Instead of saying small letter b, we don't put small letter b. There's nothing like small letter b or e. We only put o. O is a recessive form of the phenotype uh, of the phenotype allele itself. So here we don't put small letter o. We put small letter I. So the small letter I represent um the recessive trait. Um let's look at some genetic crossing. Remember, we dealt with monohybrid, and where we're looking at genetic crossing involving only one characteristic. Genetic crossing is so important. It appears in every exams. You can either gain or lose marks there, but then there are strategies you can use so that you don't lose marks. And those are the strategies I want us to adopt. You can use a line cross method or a Punnett square. I will advise we use the Punnett square because the line cross, most of you don't do it well and you end up messing everything up, losing marks. So you're going to use the Punnett square method, which is clean. And as a gentle reminder, know how to represent your alleles in exams. If it is autosomal, use the letters there. If it is gonosomal, use the X with the alleles on top. If you are dealing with blood grouping, know how to use it. You have to practice this thing to master it. So when you are given a condition like this, capital S for smooth, small s is for rough. That's how you start. You put heterozygous here, and then you put this one here, and then the first thing you do, you put P1, F1. And I'm telling you, a lot forget to do that. Just doing this will give you 1 mark. And then go to your other side, put meiosis, put fertilization here. You can even put it beside P1 and F1, it doesn't matter. And without doing anything, now you have 2 marks. Now, the genotype is not given to you except the phenotype and the key. So let's generate the genotype. Doing it will give you 1 mark. So heterozygous means it will look like this, and then it also look like that. Just doing that, we have 1 mark. Now we have 3 marks without doing anything. Now you put it this way, you separate them by the law of segregation, the law of independent assortment. Now you have your gamete. You do the same thing, segregation, independent assortment. Yes. Then what do you do? These are your gametes. What do you do with gamete? You fertilize them. So this is a line cross method. Like I said, you can do that if you are very careful, you pay attention to detail. Then what do? You list the genotype. Just listing the gamete to give you 1 mark. Listing the genotype also give you 1 mark. So already without doing anything, we have our full marks, 6 marks. It can even be 7 marks. Then the ratio, it goes on like this, and it goes on like that. And then for phenotype, yes, you group them on one side. Then you do the ratio also. You must do all of it, whether you are told to do it or not, because sometimes you learners end up ignoring some key questions and they lose mark. But if you do the whole thing, if the question demands specially that you provide the genotype aside the crossing, the genotypic ratio aside the crossing, without even paying attention to it and following the rule, you have actually answered it. You have gotten your full marks. So again, we can do the same thing using another method, P1, F1, meiosis, fertilization. You can put them side by side or you can separate them like it is on your screen. Determine the genotype now. You got 3 marks now for doing that. We produce the gametes, the law of segregation, independent assortment, distribution, that's assortment, segregation, gamete, sort it out, sort them out here. Yeah, now you're going to fertilize them. So you draw a square, then you put them here. Yes, distribute, distribute, distribute. Then what do? You cross them, you cross them, cross them, cross them. Then what do you have here? Yeah, uh-huh, uh-huh. You do your crossing. So you interpret it using the normal method. Just doing that alone gets you a mark. So this is something you need to do by practicing because it is constantly appearing in every exams. Every exams. The word is every exams. The word is every. Now let's go to blood grouping. So with blood grouping, these are the things you need to know. You need to know that the different blood groups are caused or produced by multiple alleles. So note the word multiple alleles. And then there are four blood types or four blood phenotype. They know that there are four blood types because it can be tested. You can use chemical testing to prove it that our blood types are not the same. That is why you can't donate blood to just anybody. They need to first check the blood. Apart from screening for diseases, they have to check for compatibility. So, from the research from among the human race, there are four blood types: A, B, AB, and O. And these letters you don't find it written in your blood. There are testing that you do to show, and those are physical things you can see. That's why we say there are four blood phenotypes. Four. Check the number four. The other one. These phenotypes are controlled or determined by three alleles. Behind every phenotype, there is an allele. An allele is a different form of a gene. So behind every phenotype, there is a genotype. So what is a gene causing these three letters? If you look at it, we have A, B, A, B, we have A, B, A, B, and O. There are three unique letters here. One is A, one is the B, and one is the O's. So the combination of these letters produce the four phenotype. Now, A has its own allele. We are going to see it, okay? So there are three alleles. Now, each person can inherit only two. Why two? Because it takes two to form a child. So you're going to have two letters, two alleles. One will come from your mother, one will come from your father. The alleles are your genes. Those are the things we don't see. We only see your phenotype. So when we said the phenotypes are controlled by three alleles. So let's look here. What are the alleles for A? If you check the phenotype A, B, AB, and O, we have A there. We have two types. We have A standing alone, then we have A joined to B. It is still A. So this A is controlled by an allele, which is this. This is how we represent it. Put the letter I and put the A on top. What about the B? You do the same thing. The O is represented as small letter I. Small letter I doesn't carry anything. Small letter means it's recessive. That means O is recessive, even though O is is written like a capital letter. At the genetic level, it is still recessive. So that is how we represent it. Now, these alleles, IA, IB, and I, a person can only inherit two. That is one coming from the father, one coming from the mother. So if you check your table, we have the genotype and the phenotype. So the first one is I capital A, I again capital A. That person is homozygous. Where did the person get the I capital A and the second I capital A from? The person got the first I capital A from one parent, it can be the mother. The second I came from the father. Then there's another type of blood group A where there we have one IA, the other one is I. The small letter I. The small letter I is O, but we don't put O, you just put I there. You'll be told in exams whether it's homozygous or heterozygous. That means this person is heterozygous because of the person is carrying one capital A, and then there's a recessive here. Where did the person get this alleles from? He got one from the mom, one from the dad. Same thing applies to B. What about AB? With AB, you can see both of them are dominant. Got one IA and the other one is IB. Where did the person get these two alleles from? The person got one from the mother, one from the father. Same applies to the the O. So we need to pay attention. All blood groups are caused by multiple alleles. There are four blood types: A, B, AB, and O. And these phenotypes are controlled by three alleles because there are three letters, the three unique letters in the phenotypes. And then a person can only inherit out of the three, a person can only inherit two because these are genes in our sperm and ovum. So you can inherit only two. Yes, check the numbers. Very important. So question one. Two parents, both are heterozygous for their unique blood groups or types. One is heterozygous for blood type A, the other one is heterozygous for blood type B. Using a genetic cross, what are the possible blood groups of their children? And what is the genotypic and phenotypic ratio? So use the same thing, Punnett square. We have been given the blood group. It is given to you. No marks for doing that. So you put P1, F1, you get a mark. Meiosis fertilization gives you a mark. And putting phenotype and that thing won't give you a mark because they were given to you. This will give you a mark, the genotype, because it wasn't given. So you have to determine the genotype. Heterozygous for blood type A, that means the person is going to look like this and like that because the person is heterozygous. One dominant allele, one recessive. The other one too is heterozygous. It will look like this and like that. Just putting the genotype will give you a mark. Now, let's produce the gamete. So the law of segregation, you separate them, then you put the gamete there, then you distribute, you sort the allele, independent assortment. You do the same thing here, you do the same thing here, quick, quick, quick. Then what do you do with the gametes? You fertilize them. So you do the same thing. You have to be very careful when you are dealing with blood grouping because mistakes can occur. And now here we have the gametes. Doing all these things gives you a lot of marks. So you can use another method again to do it using the same process. Whether it's blood group or sex link or autosomal, it follows the same pattern. I prefer this one, the Punnett square, because it makes the work clean and neat, avoiding unnecessary mistakes. See, the method remains the same. Everything remains the same except with blood grouping. We use the letter I. The alleles are represented differently. Okay, so that is the end of part one of the three-part series where I looked at um, monohybrid crosses and sex determination and blood groups. Okay, so the first part I was looking at genetic crossing in general, where I touched on sex inheritance and also how to do genetic crossing using the alleles in blood grouping. Now I'm going to go further into the detail to explore inheritance of sex-linked diseases and blood grouping. So I'm going to explore it to the detail. I'm going to go further into it. I've already laid a foundation, a good foundation on both. So let's check it out here. Inheritance of sex-linked diseases. So, although most of the bodily characteristics are carried on the 22 pairs of autosomes, there are few characteristics carried on the gonosomes. Certain genetic disorders are carried on the allele found on the X chromosomes only. Since these disorders are transmitted through the sex chromosomes, they are called sex-linked disorder. So take note of why we call it sex-linked disorder. Then over here in our curriculum, we're going to look at two types of disorders. The first one is going to be color blindness, and the second one will be hemophilia. And this is how their alleles are represented. You use the letter X for color blindness, you put the letter D, and for hemophilia, you put the letter H. So it is necessary to know how to go about it. Now, let's continue. Disorders transmitted through the sex chromosomes are called sex linked disorders. Color blindness and hemophilia are both caused by the recessive allele on the X chromosome. So we need to know what is the cause of this condition? No, there is a cause for every disease. So if you have a condition like this, you should know the cause. And this is caused by recessive allele located on the X chromosome. So here they are, and then that is the allele that is causing the condition. And it's a small letter. Small letter means it is recessive. We use D, small letter D, to represent it. What about hemophilia? It is a sex-linked disease. Where will you locate the disease? The disease is located on the X chromosome. Hemophilia, we use the letter H to represent the disease. It's a small letter because it is caused by a recessive allele. All right, so let's look at hemophilia. You have to know the meaning and what it is. It's the inability of the blood to clot due to lack of a blood clotting factor. This is important for terminology. What is the cause? It's caused by a recessive allele located on the X chromosome, resulting in the absence of a clotting factor in the blood. What is the effect? What's the consequence of having this recessive allele? It's going to lead to that is excessive bleeding after an injury or damage to any parts of the body. So we said the disease is caused by recessive allele on the X chromosome. So the allele is on the X chromosome. That is the allele on the X chromosome. Let's go on to some application on thinking here. Here we have to know all alleles work or function in pairs. So you're going to need two alleles to produce a phenotype. Why two? Because it takes two to produce a child. During fertilization, we need two individuals, providing a sperm and an ovum. One allele come from the father, other allele come from the mother, like XX and XY. Capital A, small letter A, capital D, small letter D. There are two, because one comes from the father, one comes from the mother. Since color blindness and hemophilia are both caused by a recessive allele on the X chromosome, hence a dominant allele also located on the X chromosomes of the gonosome prevents the expression of the disease. Note that the disease is caused by a recessive allele. That means if you have a dominant allele of that particular condition, uh, you're not gonna get it because the opposite of recessive is dominant. So if a recessive allele causes the disease, then a dominant allele won't cause it. So here we have X, and no, for it to be hemophilic, it must be recessive. Now we have another X, and this time it's normal, meaning without hemophilia. It won't give you hemophilia. That is it. So in exams, they won't give you this one. They won't give you the dominant one. They only give you the recessive. But you must use your common sense to know that if small letter H is recessive, causing the disease, then another H, which is capital letter, that is dominant, will not cause the disease. That makes you normal. Same applies to color blindness. Remember, it is caused by a recessive allele. Why are we not all having color blindness? It's because we have another allele that is actually dominant. So that is it. Okay, now let's check something here. Sex-linked disorder, hemophilia. This is how you do the genetic crossing. Hemophilia is the inability of the blood to clot due to lack of a blood clotting factor. And we're going to use the same genetic crossing method. So let's look at the situation here. So if a man is unaffected or normal, how would we represent it? That's H. You see, a man carries only one X chromosome. So either the man has it or doesn't have it. Uh, what about a carrier woman? A woman who is a carrier will look like this. She's carrying the disease genetically, but phenotypically, she's going to be normal. And then we have hemophilic man. A man who is heavily sick is going to look like this. If a woman is normal, she's purely not sick. She doesn't carry the disease. So this is how we represent it. Okay, so that is it here. Now we go to sex-linked disorder. That's hemophilia. We have to explore what is called a risk assessment. This is how we represent the phenotype. In exam, they give you only one allele, whether it's X recessive H or X dominant H. It is up for you to know if the dominant is given, know that there is another recessive. Every letter exists in pair. If it is H capital, there's going to be H small letter. Now let's look at the risk factor in males. So we have to understand that the risk factor in women and males differ. The risk factor in women inheriting this disorder is relatively low. And as you can see on your screen, there are three types of genotypic combination for female. So the phenotype is that the female can be normal or normal female with that is a carrier or hemophilic female. So for it to be hemophilic female, the probability is that you're gonna be the chance of getting it, you're gonna be actually be 1 out of 3 uh, chances of getting it. And that is why the risk factor in females appears to be relatively low, since the female will have to inherit two recessive alleles to have the disorder. And there are three ways you can produce a combination. Only 1 out of 3 will give you the condition. In the case of female, that is it. For males, there are only 2 ways of producing the combination because males are actually XY genotypically, and hence their risk factor is very high since men only one X chromosome, and as such they will have to inherit only one recessive allele to have the condition. So for men it's 50-50. Either you are normal or you're not normal, or you have the disease. So that is where the whole problem is. So let's look at sex color blindness too the same way. You have to know the meaning. A person is colorblind if he or she is unable to tell the different colors apart, for example, red and green. What is the cause? It's a sex-linked disorder. And so, like all sex-linked disorder, the condition is caused by a recessive allele, resulting in the absence of a protein, of the proteins that make up the red-green cones, that's the photoreceptors in the retina. Effect: the person will not be able to tell the difference between colors. So that is a primary effect. And specifically, the person cannot tell the difference between red and green. And so remember we said the condition is caused by recessive allele on the X chromosome. And that is it. The recessive allele is on the X chromosome. So we go on to measure the risk factor here. And it's still the same like the case of hemophilia. Uh, the risk factor in males are different from females. For females, there are 3 ways you can produce a genotype, and either the female is normal, or a female but a carrier, or the female is colorblind. For males, there are two combinations. Either you have it, either you are normal or you are colorblind. So that there's a 50-50% chance as compared to the female, where there is a 33.3% chance or 1 out of 3 chance of being normal, or 1 out of 3 of being a carrier, and 1 out of 3 of being colorblind. So the risk factor in women inheriting this disorder is relatively low since the female will have to inherit two recessive alleles to have the disorder. Whereas the risk factor in males are high since men have only one X chromosome. As such, they'll have to inherit only one recessive allele to have the condition. So question, why are males more likely to have sex-linked disorder, whether hemophilia or colorblindness? The answer remains the same. The reason is because sex-linked disorders are caused by a recessive allele. This allele is located on the X chromosome of the gonosome. Men have only one X chromosome. As such, they will have to inherit only one recessive allele to have the condition. 5 marks. So this is just simple. Now we're going to look at this question in reverse. So in reverse, now we are asking, explain why women are less likely to have sex-linked disorder. So you have to reverse the statement in the previous situation by saying sex-linked disorders are caused by a recessive allele. This allele is located on the X chromosomes of the gonosome. Women have two X chromosomes. As such, they will have to inherit two recessive alleles to have the disorder. And that is just uh, how you answer it. Now, let's go finally uh, uh, in the exploration of blood grouping. Like we earlier said, there are different blood groups caused by multiple alleles. There are 4 blood types or phenotypes, and these phenotypes are controlled by three alleles, and then a person can only inherit only two. And then we explored the alleles. They are A, B, and O. So take note of the word multiple alleles, very important. And note that there are four phenotypes, physical appearances of these blood grouping in terms of chemical testing. And these four blood groups are actually determined or controlled by three alleles because there are four blood groups, three distinct letters. So we have A, B. We don't put O there. Instead of O, we put small letter I. During fertilization, a person can inherit only the two. The alleles are in the gametes. The phenotype are the blood types by chemical reactions that we know and see through testing. So how do you inherit blood group? You inherit it through your gamete. You inherit the alleles through your gamete. And a person can only inherit two alleles, one from the sperm, one from the ovum. So you can only inherit two. So take note, it's 4, 3, 2. Four blood groups, three alleles, two two inherited alleles. Three alleles, two inherited alleles, yes. Okay, so take note, very important. Now, question, explain how to use blood groups in paternity testing. So this is how you answer it. How can you use blood group to do in paternity testing? So you say blood groups of the mother and the possible father and the child must be compared. So it takes two to form a child. So you take the mother and the child and the suspected father. Then, if the blood groups of the adults do not correspond to or match the child's blood group, then the man is not the father. So you compare the mother's blood group with that of the child. If they match, then the mother belongs to the child, or the mother is the woman is a real mother, or the child belongs to the mother. Then you compare the father's blood group with that of the child. If they match, then that man is a real father. If the blood groups of the adults correspond or match the child's blood group, then there's a possibility that the man is the father. So if you compare the man's blood group with the child and there's a match, that means that man is the father. You do the same thing for the mother. If they match, then the woman is a mother. But it is inconclusive because other testing will have to be done. Since other men may have the same blood group in many cases. Now, explain why blood grouping is not reliable for paternity testing compared to DNA profile. So let's find out why blood grouping is not reliable. Reason is because blood grouping is the reason blood grouping is not reliable is because it uses the phenotypes. It looks at the physical appearance, and behind the phenotype there's a genotype, and the phenotype is not unique or special or exclusive to the individual to trace paternity. If you are looking for your father, you can't use the phenotype. The blood grouping does not use nucleotides or genes, which is unique or special or exclusive to the individual. Other testing needs to be done since other men may have the same blood group. DNA profiling is more reliable than blood grouping since it traces paternity using the nucleotides or genes, which is unique or special to the person involved. So that's why DNA profile is more reliable. So why is DNA profile more reliable and conclusive than blood grouping? Answer is because DNA profile looks at the similarities between the nucleotide or genes in the DNA of the father and the mother. So they take the father's gene, they compare it with the child's gene. That is what is called DNA testing. If there's a match, then the man is the father. If there is no match, then the man may not be the father. The reason why they look at the similarities between the nucleotide genes is because each DNA profile is unique to an individual. 50% of the DNA fragments or bands or bars are derived or came from the mother, and 50% came from the father. That is during fertilization. Half of your genome, your DNA comes from your father, and half came from your mother. So if 50% of the DNA fragments, bands, or bars correspond or match with the father, then it can be claimed that he is the father of the child. So we just want 50% match. All right, so this marks the end of part two. Now I'll be going on to the next part, and the next part I'll be looking at pedigree. Now I'll be dealing with pedigree, where we're going to be doing an analysis and application of genetic inheritance. It's a very important concept. It appears in every exam, so you can't ignore it. So let's set the ball rolling and pedigree. All right, so first of all, we have to know the meaning of the term pedigree diagrams, also called family tree. Take note, we call it family tree or pedigree diagrams. What is it? It is a diagram used to study the inheritance of characteristics in a family over a number of generations. So let's go. Some of the things I'll skip it, some of them I won't skip it. So we have a monohybrid cross here, and what we have here are two things. We have the phenotype and the genotype. And previously we used the genotype to predict the phenotype. Over here, we are doing it in reverse. We are using the phenotype to predict the genotype. So we have two parents. One is blue, the other one is purple. One is purple, one is white. Sorry. So behind the white, there is a genotype, as the table suggested. B is dominant for blue, and small letter b is recessive for white. So if a person is white, then there must be two alleles there. Why two? Because the person got alleles from both parents. One letter B came from one parent, the other letter B came from another parent. Same applies to purple. And then you use the genotypes to do your crossing. Over here, what do we have? We have two parents. One is green, the other one is yellow. Now they produce four offspring. All of them became green. What happened to yellow? Yellow disappeared. Why did yellow disappear? It's because it was blocked by green, or yellow was masked by green. Why was it masked by green? Because green is dominant and yellow is recessive. So that is why you have it there. Yellow can only appear if the alleles are homozygous recessive. That is, we have two alleles for recessive, two recessive alleles standing together. Again, another type of crossing, observe carefully. We have two parents, one green, one yellow. Check the offspring. All of them are yellow. What happened to green? Where did green go? Why did green disappear? Green disappeared because yellow blocked it from appearing, or yellow masked the appearance or the expression or the effect of green. So why did green not appear? Green did not appear because it is recessive. It can only appear when it is in a homozygous state. Only homozygous recessive, that is two recessive alleles, can appear. So that is what we are seeing here. Another scenario, two purple parents, they produce four offspring. Three are purple, one white. Where did the white come from? Okay, to know the answer, you have to go back to the parent. If two parents of the same phenotype produce an offspring where three look like the parent and one look different, the answer is because the parents are heterozygous. So here you can use the phenotype to predict the genotype, or you can use the genotype to predict the phenotype. Look at another situation. Pink flower, white flower, they are crossed. All the offspring became pink. Where did the white go? The white disappeared because it is recessive. It was blocked by the pink color. It couldn't express itself. It can only be expressed when it is homozygous. Then you take the F1, the offspring, the all pink offspring, then you cross two of them together, pink and a pink flower. And guess what came out? You got three pink, one white. Where did the white come from? Remember at the beginning, one of the parents appeared white and disappeared in their children. The color white disappeared among their children, but has appeared in the grandchildren. That tells you, we all know white is recessive. But how can two pink parents produce three pink and one white? We don't understand, where did the white come from. If you want to know why the white appeared, you have to find out where the white got its genes from, because the white flower did not turn itself white. It became white because it was born white, and there's a gene behind it. Where did it get the genes from? If you look at the genotype, you can see it has two recessive alleles. Where did they get the two recessive alleles from? It did not buy them. It was not donated to it. It inherited the two recessive allele. One came from the father, one came from the mother. That means the parents, even though both of them were pink, they were also heterozygous. So that is how you look at it. Another situation here: purple, white, and then follow the arrow, reproduce. The offspring became purple. What happened to white? White disappeared because it is recessive. Why did purple appear? Because it is dominant. Dominant alleles are always, the word is always, not sometimes, they are always expressed. Why did white disappear? White disappeared because it was blocked or prevented or masked by the dominant allele from appearing. Then you take this offspring purple, then you cross it with another purple of the same kind, and now you have four offspring. Suddenly, the white that was blocked from appearing appears again. So, where did the white come from? It was produced, it was born white. That means it carries the genes for white. It was born from two parents that were actually purple. So, where did the white flower get its phenotype and genotype from? Definitely, it got one allele from one purple parent and the other allele from another purple parent. Take note that from the beginning, from the P1, we know white is recessive. It is only expressed when in the homozygous state. So for you to be white, you must carry two recessive allele. One came from the two purple parent. That means the purple parent are both heterozygous. So you can give it a try and generate the genotype of the following crosses. Now, let's go to the process itself, pedigree diagram. I want to finish it quickly. And we're going to use these symbols for the representation. Now you have to understand that we have the marriage line there, we have the line of descent, we have the sibling line, and then we have the generation number. So let's see how they work. So in this chart, we have a square. Remember over here, a circle represents female and a square represents a male. If it is black, it means that person is affected. If it is white, it means not affected. But you'll be given that information in exams. So let's look at the situation here. We have a family of five people. There are two generations there. The first generation represents the two parents, the square and the circle. And below them we have three individuals. One is a square, two of them are circles. So the first generation represents the two parents. The second generation represents the children or the offspring. So this family consists of five members, two of whom are males, three of whom are females. So that is two generations there. Two males, three females. And in this case too, uh, it is mostly done using either autosomal characteristics or gonosomal characteristics, or we can use the blood group in solving such a question. So you need to take note. So let's look at this genetic cross quickly. Some of them I'll just brush through because it involves a lot of repetition. So here we are. We have two parents. Both of them appear white, and then they produce three children. One appeared black. How did that occur? That is, you only have a situation like this if both parents are heterozygous for the same characteristics. If the two parents are heterozygous, then one of the offspring will be different and you need to pay attention. So the allele for white skin is dominant, and then if the allele, if white skin is controlled by dominant allele, definitely black skin will be recessive. Sometimes you need to know this on your own. So if you look at this family, only one group is black, the rest they are all white. That makes the black one a recessive trait and the rest of the white dominant. Dominant means they are more, they control the the phenotype of the family. So determine the possible genotype for the rest of the family. So this is the key. Capital D for white, small letter d for black skin. How many family members are here? Five. How many males? Two. And then how many females? Three. How many black females are here? One. How many generation? Two. That's the first two parents, the one box and the one circle. Below them is the second generation consisting of three members. So remember, for you have to set what we call the rules. For you to be white, there are two ways by which you can be white. Either you are homozygous or heterozygous. For you to be black, you just have to be recessive. So there's only one way of being black. Hence, we go to the easier one because it is easier to determine black. Since for you to be black, you must be homozygous recessive. We look into the family and determine who is black there. We check those with black skin, only one person, and that means we go to that person. That is the allele. So here is the point. This person has a black skin. Okay, he has a black skin and he has the genes for it. He has two recessive alleles and he's a product of the two parents. He's a product of the first generation. The question is, it's a she, right? Where did she get the alleles for black skin from? So the simple logic is that for you to form a child, you need two things. You need a sperm and an ovum. So this is how the person became black. She got one allele from the father, one allele from the mother. That is how she got it. So the father and the mother gave birth to the child through fertilization. So that means the father must be carrying the recessive allele for black, and the mother too is also carrying the same thing. Okay. And remember then, if you look at the parents again, they are both white. Even though they carry the black allele, they are still white. But then for you to be white, we do understand uh, for you to have a white skin, you can either be homozygous or heterozygous. But since we only know one part of the allele for the parent, that means the parents are actually heterozygous. Otherwise, you won't be able to have a child. They couldn't have such a child had they not been heterozygous. Now, it makes it easier for us to explore the other children. And in the case of the other children, there can be two possibilities for this boy here. There can be two possibilities. Either the child is like this, or if the child is homozygous or heterozygous. Same thing applies to a situation here. It can either be in this situation. So if you come across such a situation, the answer is acceptable. It's an answerable situation. Then we go to another situation here. Over here, you can see if both parents are heterozygous, again, for the same phenotype, most of the F1 will resemble the parent and a few will be different in appearance. So if you look at parent 1 and parent 2, they look alike. They had three children, one, two, three. They all look like the parent. Until three was married to a female four, and they had a child. That child looked different. And again, you can see in this family there are about seven members. There are seven members in this family. There are three generations. One and two forms one generation. One, two, three, four, they form the second generation. And then the black person at the bottom, the black box at the bottom represent the third generation. This generation consists of three females and four males. So determine the possible genotype for the rest of the family. So the allele that has been given to you, capital D for white skin, small letter d for black skin, and you set the rules. There are two ways of producing the white skin, either homozygous or heterozygous. For you to have black skin, you have only one way. That is when you are homozygous recessive. So you look for that one first because it's easy to deal with that one. So you go to the child with the black skin. You begin to trace where the child got those homozygous recessive alleles from, and you will discover that parent one and two are also heterozygous. Now let's look at how genes are inherited. So we have a man who is heterozygous for red hair, is married to a woman who is also heterozygous for red hair. So both of them have it. Their hair is red in color, and they want to form a child. If you want to form a child, the man must bring a sperm. The woman must, yeah, that's a haploid sperm there. The woman must bring a haploid ovum. And then what do? you fuse them together. That is fertilization must occur. And then what do you form? A zygote. Now you can see there are two round circles inside the zygote. The circle represents the nucleus. Inside the nucleus, that's where we have the genetic material. So that means this zygote is diploid because it has two uh, nuclei, two DNA material. One came from the father, one came from the woman. That's how it works. And so, what will be the skin appearance of the child? In this particular case, the hair appearance, the hair of the zygote or the offspring is black. How possible is that? How come a red-haired man and a red-haired woman produce a black-haired child? Does that mean the child doesn't belong to them? So we need to find out. To do so, you have been given the key, capital R for red hair, small letter R for black hair. And so we all know the child's hair is black. For you to have black hair, you must have this genotype. But how did the child get a genotype? Okay, one came from the man, one came from the woman through their gametes, sperm and ovum. That's how you transmit your genes. So, but then we all know the man's hair is already red, okay, and the woman's hair is already red. So it means already they are carrying. This man and the one and the woman they carry the allele for black hair in them. But by definition, black hair allele will never show itself in a phenotype, since it is recessive. It is always blocked by the dominant. So, but the couple, these two people, these two individuals, their hairs are red. Okay, so let's find out what their genotype will be. Remember the hair is red here. So now for that means for your hair to be red, all you need is just one dominant allele. That means the genotype for the man will be as it is, heterozygous, one capital R, one small letter r. Same thing applies to the woman. Even though she carries the recessive allele for black hair, the hair is still red. For you to have a red hair, all you need is just one dominant allele. And then there you have it. So that is how you inherit alleles. What just happened is that the offspring has inherited two recessive genes for black hair from his or her parent, one from the father and one from the mother. Okay, so we can apply the situation here in DNA profiling. The mother and the father, they have a child. Now we want to prove if the child belongs to them. So you need to check something here. So for you to form a child, you need to contribute your genes. Okay, you have to transmit your genes. This woman carries how many genes? Six genes. If you count the black bars, those bars represent the genes. And what about the father? Also carries how many? Six genes. Okay, so you count the bars, six genes. So you want to form a child. All right, the child also carries how many? Six genes. How did the child come into the world? The mother and the father produce it. The father contributed a sperm, the mother contributed an ovum. And if you remember from meiosis, an ovum and a sperm are both described as haploid. Haploid means they carry half the number of genes in the individual or half the number of chromosomes. So the total number of genes or chromosomes in a person, or that's they call it genes in a person, uh, is six. So the mother will have to contribute half. Because under traditional thinking, somebody will say, if the father and the mother contributed their genes, 6 plus 6 should have given you 12. But here, we have 6 genes from the mother, 6 genes from the father, giving us 6 genes in the child. What happened? What happened can be explained through meiosis. So during meiosis, remember, it is when a diploid cell undergo two consecutive cell division to produce four haploid cells that are dissimilar. So diploid will change to become haploid. Haploid means it has half the number of chromosomes. So if the father and the mother really gave birth to the child, let's find out how many of the genes did each contribute in the making of the child. So let's start with the child. Let's we want to let's start with the child and the mother. So let's find out from the mother how many genes did the mother contribute. That gene from the mother wasn't contributed here. Okay, this gene from the mother is also found in the child. I'll label it as M gene, mother's gene. That's the mother's gene again, found in the child. Another mom gene here. This gene from the mother is not there. This gene from the mother is in there. This gene from the mother is found in the child. That means uh, there are three genes from the mother that are present in the child. What about the father? Let's find out. This father's gene is present in the child. The father transmitted a gene. That's F gene for father gene. And then we go again. What about this gene? It's absent. What about this gene from the father? Is also present in the child. That is F gene. What about this gene from the father? Is it present in the child? Yes. So by analysis, you can tell the child also carries six genes. Why not 12? Six. Because the child was born through the natural process. Okay, gametogenesis occurred in the mother and the father to produce haploid gametes. And then when the haploid gametes fuse together, they produce a diploid gamete. Meiosis tries to uh, prevent um, abnormal number of chromosomes in an organism, ensuring constant number of chromosomes are maintained in every generation and continuity. So check the child here. The child carries six genes. Three came from the father, three came from the mother. And that is how genes are inherited. So you see here, that is it. So you can trace the genotypes that runs through a family. If we check the mother and the five children, you can tell that the five children carries part of the mother's chromosome or phenotypes. The white part represent the father, which is not captured here. If you check the one in the middle, you can actually tell that similarities in phenotypes suggest that similar genes were inherited from a common parent or ancestor. Differences may be due to a useful mutation or an adaptation to an environment. So we have a biological father and a biological mother producing a child. Can we prove that the child belongs to them? Yes, you can tell the phenotype. The colors in the child can be traced back to those two parents. Same thing applies to the mom and dad, child one and child two. You can actually tell child one and child two carries a mixture of phenotypes or genes from both parents. Um, you can trace it down the line, and we can generate a case study for pedigree. So we have this key. Watch out for this key, or you create it yourself. This is a man with red hair, a woman with red hair, and guess what? They produce a child with black hair. Now determine the genotype. So to do so, you have to set your own rules. They won't give it to you. You have to do it yourself. Here are the rules. We have dominant, homozygous dominant, two dominant R for red. There are two ways you can have red hair. Either you are homozygous dominant or heterozygous dominant. And then lastly, there's only one type of black hair produced by homozygous recessive. So you have to deal with the easier one first. It is easy to deal with the black hair because there's only one way, whereas red hair there are two ways which demands that you have to test the idea. So we don't, because black hair is easy to find, we go to the child with black hair. And over here in this family, there are two generations. That's those with red hair for on the same horizontal line is one generation. Those at the bottom, they form another generation. Why is the hair of the child black? It's because the child carries the genes for black hair. Where did the child get the genes from? There are two of them also. The child was formed by the two parents there, and he got the alleles or inherited alleles from them. So definitely, the child inherited two recessive alleles from for black hair from parents A and B. One came from A, one came from B. So that actually leads us to the final conclusion. Even though both parents carry the recessive allele for black hair, their hair is red. For you to have red hair, all you need is just one dominant allele. And you know there are two ways of having red hair. Either you are homozygous, um, either you are homozygous dominant or you are heterozygous. For you to have red hair, you just need only one. Okay, that is it. This one, and this one. Hence, the genotype for the family members are this, and you are done. And then we go to another situation here. We are dealing with noses here. That is a man with round nose and a woman with pointed nose. They produce a child. The first child was a male with round nose, and then another child, male with round nose, and a female with a pointed nose. So we have been given capital N for round nose, small letter n for pointed nose. So that's father, mother, son one, daughter, and then son two. What do you do? Determine the genotype. So you set the rule. So there are two ways of producing round nose. There are two types of round noses here, homozygous and heterozygous. That is it. So for you to have pointed nose, remember the white circle represent pointed nose, and there are two generations here. Okay, five members in a family, three males, two females. So for you to have pointed nose, all you need is just a recessive, two recessive alleles. That means the daughter's nose is pointed. That means the daughter carries two recessive alleles. And where did the child get where did the daughter get those alleles from? She inherited the two recessive alleles for pointed nose from her parent. Okay, from both parents. And then we need to find out something more. Okay, you have to trace it back. That is, one allele came from the mother, one allele came from the father. Yes, but the daughter is just like the mother. Both of them have pointed nose. That means the mother is also homozygous recessive for that trait. This leaves us with the father and the two sons. Remember, the father is carrying the recessive allele, although he has round nose. So for you to have round nose, remember there are two possibilities. Either you are homozygous dominant or heterozygous. All you need is just one dominant allele to have round nose. So because the father uh, has a round nose, that means the father's genotype is gonna be this. You see, then now that we know the genotype of the father and the mother, now we can produce the genotype for the two sons. So let's start with son one. So son one's nose is also round. That means definitely son one carries the dominant allele. Now, the mother is homozygous recessive. She can only donate one recessive allele. The father can donate either dominant or recessive, but then the child physically has a round nose. Okay, so that means the mother will donate this. The son is heterozygous like the father, but the nose is round. What about son two? Son two's nose is also round. For you to have round nose, you must have a dominant allele for round nose. And that is a contribution from the father. The mother also have to contribute something, and the mother is homozygous recessive, carrying two same alleles. So the mother's allele will look like this, and that is it. Now we have determined the um, the the genotype of the entire family members. Okay, so let's continue with this question here. So we have a large family here, and we are dealing with eye color. So first of all, we need to find out how many generations are present in this family or in this pedigree. So when we are dealing with family, we have to look at the horizontal line that puts two or more individuals together. So how many generations are here? The first generation is made up of Joshua and Ronel. The second generation consists of Sarah, Peter, and Veronica. They form one horizontal line. That's the second generation. The third generation is the third horizontal line. And there we have Malina, Frank, Jack, John, and Gail. So there are actually three generations here. Then how many males are in the family with brown eyes? How many females are in the family with blue eyes? Which phenotype is controlled by dominant allele? And determine the possible genotype of Renel, Peter, and Malena using capital B and small letter b to represent allele. So let's answer the first question. How many generations are in the pedigree? So you check here, we have one horizontally, two, thirdly, three. So Joshua and Renel forms one generation. Sarah, Peter, Veronica second generation. Malina, Frank, Jack, John, the third generation. So here there are three generations. How many males in the family? How many males are in the family with brown eyes? So you have to go to the key. So males with blue eyes are white boxes. The gray represent male with brown eyes. The circle represent females with blue eyes. Then the white circle and the gray circle represent female with brown eyes. So the question is about brown eyes for males. So how many males are born with brown eyes? So you need to check the gray color. So Peter, the box is gray, that is brown eyes, that is one. Frank, the box is gray, that is another brown eyes. So we have two. That means there are two individuals. How many females in a family have blue eyes? So let's check. Circle represent female, white circle represent blue eyes. So let's check. First generation Joshua Ronell, there's none. Sarah, second generation Sarah, Peter, Veronica. Okay, Veronica has it. So that's one there. Third generation Malina, circle, white, and there's none again. So there are again two females in a family with blue eyes. Now, which phenotype is dominant? How do you determine that? How do you determine which phenotype is dominant? You have to know what you are dealing with. So you have to check. Let's start from the top. Joshua's eyes are blue. Ronell's eyes are brown. So you have blue eyes, brown eyes, and check the offspring they produce. They had two children, Sarah and Peter, and both eyes resembles that of Ronell. Both eyes are brown. What happened to Joshua's blue eye? It disappeared in the two children. Why did it disappear? It's because it was blocked by Ronell's brown eyes, or it was masked by Ronell's brown eyes, suggesting that Ronell's brown eyes are actually controlled by a dominant allele. So the question is, which phenotype is dominant? And the answer is brown eyes. So you mix blue and white, and then you get brown. What happened to blue? It disappeared. Why did it disappear? Because brown blocked it or masked it. What do you call an allele that is able to block another allele? It's called dominant allele. Now determine the possible genotype, and they say we have to use capital B and small b for Ronell, Peter, and Malenia. So you have to know capital B is for brown, small b is for blue eyes. Then you have to know, set the rules. For you to have brown eyes, you can either be you can either be capital B, or capital B and small letter b. For you to have blue eyes, you just have to be small letter b twice to have it. So we want the genotype for Renal, Peter, and Malena. So to do so, remember Ronell, Peter, and Malena. Ronell and Peter, their eyes are brown, and Malena's eyes are blue. And we know blue is recessive. So you have to go to those ones first. So let's start with Ronell brown. Joshua is blue. Sarah is brown. Peter's eyes are brown. Okay, so let's check. Remember, Joshua's eyes are blue, okay, and blue is controlled by recessive allele. It's easy to produce it. And the father will actually provide those alleles into the children. So you need to understand something here. All right, what about Ronell? Ronell can either be like this, okay. And remember, Malena's eyes are also blue. So we know that the reason why but if you look carefully, you can see Ronald's eyes are brown and his husband Joshua's eyes are blue. That means for all the children to have brown eyes, Ronell will have to be homozygous dominant. Otherwise, if she is heterozygous, that means some of the children will have to have blue eyes. But none of the children has it. Okay, none of the children's eyes are blue, so Ronell have to be homozygous dominant. What about Malina? Malina's eyes are blue. That gives it that characteristics. That means Peter will be heterozygous because Peter will have to receive a dominant allele for brown eye from Ronell and a recessive allele for blue eyes from Joshua. But then the eyes won't turn blue because blue is recessive and brown is dominant. So Sarah too will be homozygous dominant. So we know Peter, we know Ronell, we know Malina. Oh, then we are done. What about Veronica? Eyes are blue, so that's how it's gonna be. Jack, eyes are blue. John, eyes are blue. And then what about Frank? You can tell Frank's eyes are brown, so that will make Frank heterozygous because Frank will have to receive a dominant allele from Peter and a recessive allele from Veronica. What about Gail? Okay, so Gail's eyes are brown. Gail will have to receive a dominant allele from Peter and a recessive allele from Veronica. You can actually use this mode of inheritance to predict all the genotype for the entire family member by understanding which phenotype disappear and which one remains when you mix two different colors and one disappear. That means the one that disappeared is recessive and the one that remained is dominant. So blue and then blue, 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 brown, brown, no, brown. Okay, so this one tries to explain how they got their alleles from. Okay, so let's go to another family. How many generations are here? How many males are in the family? Determine the possible genotype for person one, two, three. And I hope you are catching it by now. So let's start. How many generations are here? So you have to check again. We have one, the first horizontal line is one, second horizontal is two, the third horizontal is three. So how many generations are in this family? Three. How many males are in the family? So you have to count. Okay. So a box represents a male. That is one, two, three, four, five. So five. Now determine the possible genotype of one, two, and then seven. So remember the key suggests black is for normal, black circle is normal female, white circle albino female. Black um black square is normal female, white square is an albino female. So one one is an albino. Okay, remember you have to use N represent normal skin. Small letter capital N is for normal skin. Small letter N represent an albino. So for it to be an albino, you set the rules. This is for normal skin. This is also for normal skin. This is for an albino. Wait a minute, that's incorrect. That's a mistake there. Let me correct it quickly. Okay. All right, so let's look at another situation here, like we were doing earlier on. So, how many generations are present in this diagram, pedigree diagram? How many males are in the family? Determine the possible genotype for person 1, 2, and 7. If N represent normal, capital N represent normal skin of color, and small letter N represent albino. So this is how it goes. For you to determine the generation, you do it horizontally. So 1 and 2, they form the first generation. 3, 4, 5, 6, 7 forms the second generation. 8, 9, 10, 11 forms the last generation. So over here, we have 1, 2, 3 generations. So the answer is going to be 3. How many males are in the family? Go to the key. A circle represents a female. Black circle is normal. White circle is an albino. Black square is normal, and the white square is a normal male and a normal female. So we check how many males are here. 1 is a male, 3 is a male, 7 is a male, that's 3 males, 8 is a male, and then 9 is also what? A male. So that is 5. So we have 5 males. Now determine the possible genotype of person 1, 2, 7. If N represents normal skin and small capital N is normal skin, small letter N represents albino. So we set the rules. Here are the rules. We have been told what we are dealing with. For you to have normal skin, we know N is dominant over uh, N is dominant over small letter, capital N is dominant over small letter N. So for you to have normal skin, you need a dominant allele. And then albino skin, you need a recessive allele. So there are two ways of producing a normal skin, but there's only one way of producing albino. So let's go to the albino because albino represent the white box. Let's deal with albinos first. So 1 is an albino and 2, okay, you can see 2 is not an albino. But what kind of normal skin would this person be? Will the person be homozygous or hetero? Over here, the person's skin is genotype. The person's skin is normal, controlled by a homozygous dominant allele. Why? Check 3, 4, 5, 6. All of them, their skin appeared to be normal. What happened to um, the recessive allele carried by 1? They were blocked by the dominant allele carried by 2. If 2 was heterozygous, some of the children's skin would have been albino. But as you can see, 3, 4, 5, 6, their skins are all normal, perfectly normal, suggesting that 2 is actually carrying a homozygous dominant allele. Yeah, so this is going to be like that. 3 is like this. What about 4? What about 5? What about 6? For them all to have the black skin, they are supposed to have this kind of genotype. That means 3 has 3 has two genotypes. One came from the father, one came from the mother. The capital N came from the mother, the small letter n came from the father. You can trace it back. 4 to the same. You see, you can trace 5. The capital came from the mother, and the small letter came from the father, who is an albino, and it carries on. Then 6 was married to 7. Both of them had black skin, but check their children. 8 skin was black. 8 has a black skin. 9 is albino. 10 and 11, they are both normal female. So the question is, if two parents, 6 and 7, are normal for skin color, how come they produce such a child? So first of all, you have to know that 7 can either be heterozygous, okay, or homozygous. So that can bring a little bit of confusion. But this confusion can be cleared by looking at the offspring with a recessive allele. There are 3 of them, 8, 9. There are 4 of them, 8, 9, 10, 11. 3 are normal, 1 is albino. So you deal with the easier one first because there is only one way of producing an albino skin. And 9 is an albino. So 9's genotype will appear this way. Then you ask yourself, where did 9 get those two alleles from? The two recessive alleles from? You will discover that one allele came from 6, one came from 7, because they gave birth to 9. You can't get what your parent did not give you. You inherit whatever you have from your parent. So now, if 9 carries two recessive allele, or if 9 is homozygous for recessive trait, that means 9 got one allele from the mother and one from the father, whether they like it or not. They gave the child the alleles. You see? So that means 6, we all know, is heterozygous. That means 7, we can avoid the confusion by saying heterozygous or homozygous. Now we know what 7 will be, because 7 contributed to the formation of child 9, meaning 7 is heterozygous. Definitively, we can confirm from the child. You can use the child to determine the parent's genotype, or you can use the parent to determine the child's genotype. You can't have what your parent did not give you. And whatever you have, you got it from your parent. So for this child, he got his alleles from the two, 6 and 7, from the two parents, 6 and 7. 7 donated a recessive, and A6 also donated a recessive. Otherwise, he wouldn't have been albino. What happened to the rest? Now, the rest, it can be hypothetical. For example, 8 can be 8 can be homozygous dominant or heterozygous. 10 too can also be homozygous or heterozygous. And 11 too can be homozygous or heterozygous. But if you follow the normal Mendelian genetics, you can assume 8 is homozygous dominant and the rest will be heterozygous. So this question is incomplete. Then we go to this one also. How many generations are represented here? So we count the horizontal lines: 1, 2, 3. So there are 3 generations here. How many males are in the family? So let's check. The box represents the male. That is, first generation we have 1, second generation we have 2, 3, 4, 5, 6, 7, 8 in a third. So there are 8 family members. Determine the possible genotype of person 1, 2, and 7. Now S represents straight hair. The small letter S represents curly hair. So again, you set the rules. So these are the rules of the house, or the rules of the game. So you set the rules of the game here. There are 2 ways of having straight hair. You can be homozygous for straight hair, or you can be heterozygous for straight hair. But for you to have a curly hair, you have only one way. That is being homozygous recessive. So that means the question is incomplete anyway. Is there are some missing links here, a lot of them. But you can determine which phenotype is recessive, which phenotype is dominant. So here, if you start from the top, a female with straight hair and a male with straight hair produce four offspring: two females, two males, three three have straight hair, and one has uh, a curly hair. That means curly hair is recessive and straight hair is dominant. So first of all, one will have to be recessive or homozygous recessive or homozygous dominant. What about female? The female too, the same. So this is a real hypothetical question. But then we know what two is because we're answering two, three. Why is one homozygous recessive? Because if both if both parent here, that is the female and the male, if they're not heterozygous, they won't be able to have this child. But this child is recessive, carrying a recessive allele, and for the child to be recessive, he must receive a recessive allele from one parent here and another recessive allele from another parent. That means both of them are recessive. Now check these children here. This man is this man's hair is straight. This woman's hair is also straight. All the children's hair are straight. And we know this female here has a straight hair because of the appearance. Okay. And that means for all their children to have straight hair, none of the parents should be heterozygous. That means one parent can be heterozygous, the other parent will have to be homozygous so that all their children can have straight hair. Seven is recessive. As it appears, we have two dominant parent with straight hair producing three offspring. One has a curly hair. Where did she get a curly hair from? I mean, that means both parents are heterozygous. They were carrying the recessive allele that appeared in within the children. Okay, so hemophilia 2 is connected in pedigree. So let's look at it here. You must know that the gonosomes for male is mostly XY, one large chromosome, one small chromosome. They are unidentical. The gonosomes for females are identical, both large gonosomes. So let's look at the family here. Hemophilia is caused by a recessive allele on the X chromosome. That is it. Now, here is the key. The white square is normal. The white circle is female normal. Black square is hemophilic male. The black circle is hemophilic female. So let's check here. We have a family here. One, a male married to a female. Both of them are normal. They produce first child, male, normal. Second child, normal. Third child, hemophilic male. He is married to a female who is normal, and they produce two children, a normal female and a normal male. Okay, what are you supposed to do? Determine the genotype of all of them. Have you seen it? Okay, so already we have been given one allele, that is the hemophilia allele, which is X recessive H. Okay, so we set the rules and we know this one is dominant. Remember, the question said the small letter h is recessive, so that means the capital H is dominant. All right, so we have to fix the gender for all of them. This male, 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 male. And now we need to use the key to determine, because a male can carry only one X. So a male can either be normal um or hemophilic. Either the male is homozygous or uh, recessive or dominant. So let's determine which of the males. There are four males here, three generations, four males, four female. How many males are hemophilic? How many are normal? So let's start. One, the first generation male is normal. The second generation, there's a normal here. Then the third generation, there's a normal here. Very good. And then we have the individual 3 who is hemophilic. So that is it. We already got the alleles for 1, 4, 3, and 7. Then let's go to the other one. Let's find out, because remember it was 1 and 2 that produced this individual. And we all know for it to be a male, you must have the Y chromosome. Okay, because without the Y chromosome, you can't be a male. That means the Y chromosome that V carries, he inherited it from one, from the father. The father gave the child the Y. That's why the person is a male. Now, males are XY. So if the Y came from the father, the X will have to come from the mother. So that means the mother carries a normal allele. That's why the mother is actually normal. All right. But we all know women are XX. Okay. All right. Now let's go to the second boy in the family. Son number 3 is a male, but it's hemophilic. The boy got a Y from the father, because you can't be a male without a Y. For you to be a Y, only the man can give you the Y. So the Y came from the mother. But male are XY. So if the Y came from the father, where did the X recessive H come from? This came from the mother. That means the mother is a carrier. So now we know the genotype of the mother. That means we cannot predict the genotype of daughter V. That's the daughter there. So females are XX, and she's normal. So definitely she got one normal allele from the father. Now the mother can donate any two, because the child there is normal. So either the mother gave the child this, or the child can still receive the normal allele from the mother, from the father, and receive a recessive allele from the mother, which will still make the child normal. So that is what those are the technicalities you need to know. We are dealing with gender. Sex-linked disease means gender-based disease. So that is it. Now let's go to um 6, 7. Now 3 is married to 7. 3 married to 6, and they produce 2 children. 6. Oh, all right. That's a little mistake there. That's 2 children, 1 boy, 1 girl. But we know the gender for the boy. The boy is normal. And for it to be a boy, you need the Y allele from your father. So the father donated the Y allele. Differently, the X, the normal recessive, the dominant recessive or the normal recessive allele will come from the mother. That is 6. Okay, VI. Okay, so at least we know half of the story for VI. And now this individual here is normal. Now for it to be normal, remember she's a female, so she must carry 2 X chromosome. That means the father will donate one, because the father is hemophilic. She will donate 1 X chromosome, which is recessive for the disease, which causes the disease. But then the girl is normal. That means she will have to inherit a recessive, another dominant allele from the. Now that is again hypothetical in nature, because VI can also be heterozygous. So, but at least we can still accept it the way it is. The aim is to build understanding in the concept. So you can see the females. That is to say, this female is similar to this female. That is the point. Uh, because their characteristic, they can either be normal or a carrier, and phenotypically they will still be normal. So let's look at this one also. How many generations are represented in the pedigree? So you check again, horizontally count it: 1, 2, 3, 4. So there are 4 of them. How many males are in the family? So let's check. A square is a male, either normal or hemophilic. So we have 1. In the first generation, we have 1. Second generation, we have 1, 2, 3, 4. Third generation, we have 5, 6, 7. Fourth generation, we have 8, 9. So in all, we have 1, 2, 3, 4, 5, 6, 7, 8, 9. How many females are hemophilic? So let's check. When you are a circle, you are a female, but when you are white circle, you are normal. Gray circle, you are hemophilic. So how many hemophilic? First generation, no hemophilic female. Second generation, hemophilic is here. And so that gives us only 1. Now determine the possible genotype of person 1, 2, and 7. So you have to set the rules. Either you are like this or you are like that. Okay, and then you can actually work it out. You can give it a try and try and play around it. All right, so this brings us to the end of the whole concepts. Beautifully done. I will advise you to read over this thing. You have to go over it left and right. You don't just study it once and you say, "No, I'm done." No, it doesn't work like that. You have to repeat it. It is necessary to listen to it. I've modified the video to help you to listen, watch, and see how things are done. Remember, practice makes a man perfect. The best way to learn something is to do it, and the best way to remember something is to repeat it. So, if you want to know something, do it. If you want to remember something, repeat it. And you have to, if you continue doing that, everything becomes perfect for you. Thank you very much for